So our project starts as usual with a challenge. Our friend Sarah, a postdoc in the lab, has been tasked with finding new genes that interact with ATR or that are related to ATR, and she wants to use the DepMap dataset for that. And her idea is that she can find genes that have a similar pattern of dependency that correlate with ATR in their gene dependency pattern, and she will compare this in the breast and the myeloid cancer patterns. For this, she'll do a proper data analysis, a data mining project, and generate lots of visualizations and use lots of statistical tools. And hopefully this will give us new insights into find new genes that potentially interact or work together with ATR. To understand this, and this is probably the most important part of this lecture here, is to really understand the concept of what we're trying to achieve here. Um, these type of gene dependency correlations are a really powerful source of information from the DepMap data. So what we have is for each gene, we have data for hundreds of different cell lines um, that tell us how important is that gene in that cell line. So if we plot two genes on a scatter plot, we can align the cell lines here. Each dot is a cell line, and then we can see, so what is the for this cell line here, for example, let's say this is HeLa cells. They are very sensitive to ATRP, and they're also very sensitive to ATR. And that will immediately tell us whether there's a pattern or not. So if the cell lines fall onto this nice linear line, so if they form this linear structure here, that means that cells that are dependent on ATR are also dependent on ATRP. So ATR is almost a predictor for ATRP dependency. And cells that are independent of ATR are also independent of ATRP. And ATRP and ATR work together. So that would be a positive control almost. If that's the case, we can conclude that these two genes must somehow be functionally related. And in the case of ATRP and ATR, they actually are in a complex together. So we know that already. But in this way, we can check all the 17,000 genes and find new genes that have a similar pattern, that fall into a linear correlation in their gene dependencies. There's also the possibility that you have negative correlation, which is slightly more complicated to interpret, but it also tells you that something interesting is going on. So if here, for example, this gene is not dependent on ATR, but it is very dependent on ATR. So it's here on the left. So it's in the high dependency region for ATR, but it's not at all essential for this gene here for MDM2. Whereas genes that are more dependent on MDM2 are less dependent on ATR. So there's an inverse trend here. Most genes will fall into this pattern here where you have no correlation and you have just a random pattern. So we need to find statistical tests that tell us how linear is this correlation, how much correlation do we have, and then find the top hits and then do a lot of statistical analysis to verify, validate these things. This will be the first thing we do. So our workflow, so before we do that, let's talk a little bit about the biological context. ATR is a really critical gene. It's a guardian of the genome, and it gets activated when DNA is broken and damaged. And it is a kinase, and it then activates loads of downstream regulators to initiate DNA repair, to initiate a checkpoint, to halt the cell cycle. So it's a critical gene. Cancer cells have a lot of DNA damage, so they're often very dependent on ATR, so it's an interesting drug target as well. And it's a highly essential gene, so it's quite an interesting gene to study. And also it's highly relevant in the Genome Center because it's been first described in fission yeast by Tony Carr in the center. Right, so let's go through our workflow. So this is the program for today. We start off with exploratory data analysis, and we've done most of that, so I will go through relatively quickly. There's one missing thing that we need to do is like what to do with missing values, so we'll talk about that. Then we need to do a huge analysis on looking at correlations between ATR and all the other genes. So we have to do this correlation analysis 17,000 times and then calculate Pearson and Spearman correlation and p-values. And that will be then our result. And that we have to statistically verify. So we do false discovery ratios and analyze this to get rid of false positives. And then we repeat the whole analysis again with our top gene that we identify. So we have two sets of genes from two independent genes that with our query gene and the gene that we identify. And then we see whether there's an overlap in these gene sets. And that's in this network analysis that we do at the end. So quite a program to go through. And I've given you the code for this end-to-end analysis in a notebook, so you can go through that. We go through that slowly in the part of this course. So as usual, we import all our packages, and then we import the data, and then we get going with the individual steps. And we'll do that in the next lecture.